Welcome back to the Deep Dive. Today we are tackling a subject that feels a bit like science fiction, but it's actually solving what might be the single biggest headache in modern biology. We are talking about the holy grail of cellular mapping.
It really is the holy grail. For the last few years, we've had this massive disconnect. We have incredible tools to see what our cells are doing and separate tools to see where they are, but putting them together has been a nightmare.
Right. To set the stage for everyone listening, I want to use the city census analogy because it really helps click this into place. Imagine you have a list of every single person in New York City. You know their names, their jobs, baker, banker, bus driver, and you know their hobbies. That list is your who. In biology, that's single-cell RNA sequencing.
Exactly. It's a parts list, high detail, but zero context. You have the list, but you don't know where anyone lives.
And then you have satellite photo of New York at night. You can see the lights, the neighborhoods, the traffic flow. You know where the energy is. That's your where. In biology, that's spatial transcriptomics. But here's the problem. The satellite photo is blurry. You see a blob of light, but you can't tell if that's the banker or the baker living there.
Precisely. And for years, researchers have been trying to just, smash these two data sets together. They try to guess which cell from the list belongs to which blob on the map. And usually it's a mess, it's imprecise, it's noisy, and you end up losing a lot of data.
Until now. Or at least that's the promise of the paper we are diving into today. It was published in Nature Communications in July 2025, and it proposes a new method called CMAP.
Cellular mapping of attributes with position.
CMAP. The claim here is that they can mathematically fuse these two data types to give every single cell a GPS tracker exact coordinates. We're going to unpack the three levels of how this algorithm works, touching on some wild physics, and then look at two specific case studies, kidneys and cancer, where this map changed what we thought we knew about biology.
And what I love about this paper is the strategy. They call it divide and conquer. They realized you can't just throw 20,000 cells at a tissue map and hope they stick. You have to break the problem down into manageable chunks.
So let's get into the mechanics. How do you map 20,000 individual cells to thousands of blurry spots so the computer doesn't just, melt?
You do it in three distinct tiers. Level 1 is domain division, level 2 is optimal spot, and level 3, which I think is the real innovation, is precise location.
Okay, let's unpack level 1, domain division. This sounds like what, the first step in sorting mail?
That's a perfect way to visualize it. Before CMA tries to find the specific house a cell belongs to, it first has to figure out the zip code. It doesn't treat the tissue as one big chaotic block. It uses an algorithm called HMRF, Hidden Markov Random Field.
HMRF. Okay, that's a mouthful. What is it actually doing?
Think of it as a neighborhood scout. It looks at the spatial data, the blurry map, and it identifies coherent patterns. It says, okay, looking at the gene expression here, this area is clearly the tumor core, or this area over here is the cortex. It draws boundaries around these regions to define spatial domains.
So it carves the map into neighborhoods.
Correct. And once those neighborhoods are defined, CMAP looks at your single-cell list, the census. It identifies genes that are specific to those neighborhoods, and then it trains a classifier, specifically a support vector machine, or SVM.
I remember SVMs from Data Science 101. They're kind of like a bouncer at a club deciding who gets in and who doesn't.
Exactly. Or the sorting hat from Harry Potter. You feed it a cell and it asks, based on your genes, do you look like you belong in the tumor core or the cortex? And assigns the cell to a domain. But, and this is critical, it assigns a probability score.
A confidence score.
Right. And they set a hard filter. If the probability is too low, say, below 0.5, CMAP just says, I'm not sure where you belong, and it discards the cell.
It seems harsh. Why throw data away?
Because in real experiments, you have noise. You have free-floating RNA, dead cells, or just junk data. If you try to force map a junk cell, you pollute your entire map. This step ensures that we are only mapping the cells that actually belong there.
Okay, so that's level 1. We've filtered the junk and sorted the cells into zip codes. Now we move to level 2, optimal spot. We're in the right neighborhood, but we need to find this specific house.
Or the specific voxel or capture spot on the slide. Yeah. This is where the math gets really elegant. Within that domain, CMA generates a random alignment matrix. It basically just starts trying to line up the cells with the spots.
But how does it know if the alignment is any good?
It uses a cost function optimization. It's trying to minimize the difference between the cell and the spot. But unlike other methods that just, count genes like beans, CMA uses a metric from image processing called SSIM.
SSIM. Structural Similarity Index. Isn't that what they use to compress JPEGs and things?
Exactly. This is brilliant because SM doesn't just look at absolute numbers, it looks at the texture. It looks at luminance, contrast, and structural patterns. It ensures that the pattern of gene expression in the cell matches the pattern in the spot.
Huh. So it's matching the vibe or the shape of the data, not just the raw count.
Yes. And they pair that with a second metric called information entropy.
Which sounds like chaos theory, but here I'm guessing it's about crowd control.
Spot on. Without entropy, an algorithm might get lazy. It might find one spot that looks perfect and just dump 500 cells into it. Information entropy acts as a penalty for overcrowding. It forces the algorithm to distribute the cells realistically to mimic the actual density of biological tissue.
So SM ensures the match looks right, and entropy ensures the cells have some personal space.
Correct. And to solve this massive equation, they use an optimizer called Needham-Nesterov Accelerated Adaptive Moment Estimation. It's a gradient descent algorithm, kind of like what powers deep learning. It just iterates over and over until it finds the optimal spot for every single cell.
Okay, before we get to the physics part, let's do a quick recap. It's been about 5 minutes, and I want to make sure we have this workflow locked in.
Good idea. Let's summarize.
So, step 1 is domain division. The goal is sorting the mail. We use HMRF to identify spatial neighborhoods and a support vector machine, SVM, to sort cells into those neighborhoods, filtering out the low-confidence junk.
Check.
Step 2 is optimal spot. The goal here is to find the house. We use SM to match the structural pattern of the genes, the texture and information entropy to ensure realistic density. And the Needham algorithm runs all the calculations to lock the cells into specific capture spots.
Perfect summary. But we still have a problem. Those capture spots, they're big. In a technology like Visium, a spot is 50 microns wide. A single cell is maybe 10 or 15 microns.
So saying a cell is in the spot is like saying I'm in the football stadium. It doesn't tell me which seat I'm in.
Exactly. And that brings us to level 3, precise location. This is where they stop treating it like statistics and start treating it like physics.
The spring steady state model. This was my favorite part of the paper.
It's so incredibly visual. Imagine you have a cell and you've assigned it to a spot, but you also know who the neighbors of that spot are, up, down, left, and right.
Okay, I'm visualizing a grid.
CMA mathematically attaches springs from the cell to its assigned spot and also to all the surrounding neighbor spots. Now, these aren't normal springs. The tension or the stiffness of each spring is determined by the Spearman rank correlation coefficient.
Let's unpack that. Spearman correlation measures similarity. So if my cell is basically genetic twins with the left neighbor spot, what happens?
The spring connecting it to the left becomes very stiff and very strong. It pulls hard. If the cell is totally different from the right neighbor, that spring is loose and weak. It barely pulls at all.
So it becomes a microscopic game of tug of war.
Precisely. The algorithm solves a force decomposition equation to find equilibrium, the point where the net force is 0. If the cell is really, really similar to the left neighbor, the left spring wins the tug of war, and the cell physically slides over to the left side of its assigned spot.
That is genius. It's using the neighbors to triangulate the exact x-coordinate between the grid points.
It reconstructs the biological continuum. It's no longer a pixelated grid, it's a fluid, high-resolution map.
But does it actually work, or is this just some fancy math on a whiteboard?
Oh, they benchmarked it heavily. They ran a simulation on the mouse olfactory bulb, the MOB, and compared CMAP to the leading competitors, CellTrek and Cytospace.
And what was the verdict?
2 things just jumped out. First, cell usage ratio. CMAP mapped 99% of the original cells.
99%. And the others?
CellTrek lost 55% of the data. Cytospace lost 48%. They essentially threw away half the census because they couldn't confidently place the cells.
That is huge. If I'm a researcher, I paid good money for those cells. I do not want an algorithm that just tosses half of them in the trash.
Exactly. And the second verdict was visual accuracy. They used a technology called seq-o-esh as a ground truth. Because it's imaging-based, we know exactly where the cells are supposed to be. CMPOP reconstructed the shape of the tissue layers perfectly. The competitors, they produced these fuzzy, dispersed maps that looked like static on an old TV.
And they even went up against Xenium, which is the newest, highest-res tech, right?
Yes. And CMPOP accurately reconstructed these incredibly fine structures, like the boundaries of DCIS, ductal carcinoma in situ. It could tell the difference between the tumor cells and the myoepithelial layer wrapping around them. The other methods just blurred those lines together.
So level 3 uses a spring model physics to pull cells toward the neighbors they resemble, fixing their exact coordinates. And the benchmarking shows it just flat out beats the competition by keeping more data and creating sharper maps.
That's the technical victory, but the reason we care is the biology. Better maps mean better science.
Right. Let's talk about the biological wins. The first case study is about endothelial cells.
This was fascinating. So endothelial cells, or ECs, are the cells that line your blood vessels. And historically, in single-cell data, they all look kind of the same. An endothelial cell from the kidney looks a lot like one from the stomach. They're just, the plumbing.
Pipes are pipes, essentially.
That's the assumption. But they used CMAP on a data set of a mouse embryo. They mapped these generic-looking endothelial cells onto a special map of the visceral organs, kidney, gonad, stomach.
And because CMAP places them so precisely, they could ask, what makes the kidney pipe different from the stomach pipe?
Exactly. And they found these hidden organ-specific identities. For the metanephros, which becomes the kidney CMAP, identified a specific gene called DUT.
D-U-T.
Right. For the gonads, it was the gene CTSL. And for the stomach, it was GALT18.
And these weren't known before.
Not as definitive spatial markers at this stage, no.
Right.
Standard analysis missed them because without the spatial context, the signal was just too weak. But here's the kicker. When they looked at the competitor, CellTrek, it scattered these cells randomly.
So CellTrek was basically saying, I don't know, maybe put the kidney pipe in the stomach.
Pretty much. It missed the pattern entirely. CMA put the DUT-positive cells right in the kidney region. And they validated this with RNA in situ hybridization, which is like taking a picture of the RNA in the real tissue. Sure enough, the kidney vessels lit up with DUT.
So the plumbing actually has a local flavor depending on which organ it's in. That's incredible.
It proves that location drives identity.
Speaking of location driving identity, let's move to the second case study, the tumor microenvironment. This is the battlefield where immune cells meet cancer.
This is where the resolution of that spring model really shines. They were analyzing lung cancer patient data. Specifically, they were looking for something called TLS tertiary lymphoid structures.
They're like pop-up command centers for the immune system inside the tumor, right?
Exactly. It's where T cells and B cells get together to coordinate the attack. Because CMA can place cells in the gaps between the spots, it allowed the researchers to measure the exact distance between individual T cells and B cells.
So they could define near and far mathematically.
Yes. They classified T cells as T-near if they were within 10 microns of a B cell. I mean, they're basically touching. And T-far if they were more than 200 microns away.
And what happens when a T cell cuddles up to a B cell?
They found a molecular transformation. The T-near cells express a signature of TFHT follicular helper cells. Genes like BHL-HE40, TOX, ICOS, these are genes that basically say, I am now helping the B cells make antibodies to fight this thing.
And the 2,4 cells.
They didn't have this signature. This implies that the physical proximity, the hug, is what drives the differentiation, and they even worked out the mechanism.
A handshake.
Right. It involves a CD40LG gene on the T cell binding to the CD40 receptor on the B cell. And vice versa, the B cell uses TNF-SF9 to bind to the T cell's TNFR-SF9. It's a two-way conversation. The B cells are effectively coaching the T cells to fight the tumor.
Okay, now here's the scary part. What did the other algorithms show?
CellTrek actually showed the opposite pattern. It showed a decreasing score with proximity.
Wait. So if I use the other map, I would think the B cells are turning the T cells off.
Potentially, yes. Because CellTrek was randomly scattering cells near the spots, it blurred the signal so badly that it actually inverted the biological correlation.
That is a stark reminder. A bad map isn't just ugly, it's dangerous. It leads to false biological conclusions.
Absolutely. Accurate mapping is critical for biological truth.
Let's do our second recap here, covering the physics and the biology we just discussed.
Let's lock it in.
Level 3 is precise location. We use a spring steady-state model where the tension is based on Spearman correlation. The cell is pulled by its neighbors to find an equilibrium point, giving a subspot resolution.
Correct.
Application 1, endothelial cells. CY-PRE revealed these organ-specific markers like DUT in the kidney and C2LA in the gonad that tell us blood vessels adapt to their organs. Competitors miss this completely. And application 2, cancer immunology. CMAP allowed for distance-based analysis in lung cancer. It showed that T-near cells, the ones close to B cells, transform into helper cells via the CD40-LG CD40 pathway. Competitors got the relationship backwards.
That's the full picture.
So we've praised CMAP a lot, but you're the expert. You have to tell me what's wrong with it. What are the limitations?
Well, yes, CMA isn't magic. It relies entirely on the quality of the pairing. We call this the reference matching problem. If your single-cell data doesn't match your tissue type, say, you try to map a brain cell census onto a liver slice, it's garbage in, garbage out. The algorithm will try to find a match, but it will be total nonsense.
So you need to make sure your census actually matches your city.
Exactly. Also, estimating the exact number of cells per spot is still really hard. Just because you have a lot of RNA counts doesn't always mean you have a lot of cells. A big cell might have more RNA than 3 small cells. CMAT uses that entropy metric to try to balance it, but it's still an estimation, not a photograph.
And looking forward, where does this go? What's the next level?
Multi-omics. Right now we're mapping RNA. Imagine mapping epigenetics, the software of the genome, and proteins at the same time, with this level of spatial precision. That's the dream, integrating who, where, and what state all in one map.
I want to leave the listeners with a thought that really struck me while reading about the spring model. We talked about how the algorithm solves for equilibrium, where the net force of all those springs is 0.
Right. It stabilizes the position.
It makes me wonder, is a healthy tissue just a mathematical equation that equals 0? Is health simply the state where cellular tension is in perfect balance?
That is a profound way to look at it.
Right.
It implies that disease like cancer metastasis might effectively be a disruption of that tension, a failure of the forces to hold the structure together. If the springs break, the cells wander.
Exactly. So for everyone listening, here's a question for you to chew on. If you could use CMAP to map any tissue in the human body to see exactly who is talking to whom, which medical mystery would you try to solve first?
That's a tough one.
Would you map the brain to find the broken circuits in Alzheimer's, to see which neurons stop talking to each other? Or would you map the immune system to see why it turns on itself in autoimmunity?
Both rely on conversations we just haven't been able to hear clearly. Until now.
Thanks for taking this deep dive with us. Keep your coordinates precise, and we'll see you next time.